This is a conversation with Jeff Coller. He's a professor at the Johns Hopkins University School of Medicine, where his lab studies RNA biology, specifically how mRNA stability, decay, and translation regulates gene expression. In this conversation, we talk about how RNA emerged as an active regulatory layer in biology, the recent hype for RNA therapeutics, and what it's like to ask precise questions in a field that's constantly evolving. This is a Acc Podcast. Please enjoy. Welcome, Professor Coller, to the podcast. It's really great to have you. So you entered RNA biology quite a bit of time ago, right? In the 1990s, well before this current boom, and you trained in 2 HHMI labs that sort of focused on fundamental mechanisms. I'm curious about just what drew you to RNA back then, because right now it's super hot, right? But back then, I'm sure it was not. And yeah, what drew you to the field?
It really was sort of at the time in the 1990s when I was an undergrad, I was being told as an undergrad that, all enzymes are proteins, but not all proteins are enzymes. And that was like the dogma of the day, is that there's no such thing as an enzyme that's not a protein. And I had the opportunity to go abroad and study in the molecular biology department in Stockholm University. And I started working on these things called group 1 introns. And these were found in bacteriophage, so specifically in bacteriophage T4. And I was looking at these and realized that they are enzymes. I didn't realize it, but that it had been discovered just a few years prior that these group 1 introns were self-splicing. And Tom Cech and Sidney Altman had won the Nobel Prize for that a few years prior to when I was in Stockholm. And I thought that was absolutely fascinating that, here is this fundamental change in the way we think about biology, that RNA can be an enzyme. And, all of my professors were wrong when they were telling me that, enzymes are always proteins. And so that disconnect between what I was being taught through textbooks and what I was seeing in the real world in research was really exciting, and I was hooked.
Yeah, group 1 introns are ribozymes,
That's correct. Yeah,
right?
that's correct.
Back then, RNA enzymes, I'm sure that concept was pretty novel. Was there any skepticism or pushback from people in your department or people in the field, or was it?
Yeah.
Did it just
Well,
take some
no,
time
I
to
mean, it
get
wasn't
there?
necessarily pushback from what I was working on because it was generally well accepted by the time, this is about 1993, that RNA could have catalytic activity. The story of how Tom Cech and Sid Altman got that discovery, of course, had a lot of, ups and downs, and they had to do a lot of experiments to prove that. But it wasn't necessarily that what I was seeing was at all, controversial at that time. But the thing that really got me excited about RNA more than anything. So when I finished that work in Stockholm, I went to graduate school and I wanted to go to where there were a lot of RNA researchers. And so there was about 2 or 3 places in the world, in the United States at least, that there were a lot of RNA researchers. There was Yale and there was the University of Wisconsin-Madison. And oddly enough, the University of Wisconsin-Madison had like the highest concentration of RNA researchers in the world at that time. So I went there for graduate school and I got really excited about early embryonic development because what I found that was really fascinating at the time we understood about what we call maternal mRNAs. And these are mRNAs that are expressed in the early embryo or early oocyte and almost every mRNA is expressed. And basically, the first developmental decisions that are made upon maturation of an oocyte into an egg, as well as the first decisions in embryogenesis, are all made by turning on and off these maternal mRNAs in terms of their expression. There's very little transcription, if any, in the early embryo. And so the way in which you fire all these mRNAs and then mask them is through polyadenylation of their 3' end. And so you add a poly A tail, you stimulate its translation, you remove a poly A tail, you block its translation. And what this really meant was that even after fertilization, the paternal genome was really quite irrelevant for early developmental decisions. It was all the mother's mRNA. And so I thought that was really so cool because it means like your mother was the most important thing in your life from the beginning, right? And up until mid-blastular transition. And I found that really exciting. That's what got me hooked on really studying mRNA metabolism, and I haven't switched directions ever since.
I feel like that's such an interesting, not just like an interesting scientific discovery, but it's an interesting narrative that I think a lot of people probably are not aware of how much. I mean, the paternal Y chromosome, right, is super super
Yeah.
small. It barely contains any genes, and I think people outside of the scientific community don't realize how much of that early embryogenesis is influenced by maternal DNA. So that, I mean, it's a cool scientific discovery nonetheless, but also an interesting
Yeah.
narrative that I think more people should know
Oh
about.
yeah.
I mean, the timing, these maternal mRNAs are like highly regulated, right?
Yeah.
That's my
Hmm.
knowledge. They're stored, translated, I mean, and degraded with very almost exquisite
That's right.
timing. Was it the coordination of those layers that also kind of fascinated you because
Yeah.
you go on to do research that very yeah,
Yeah, because at the
go for it.
time, nobody really understood. I mean, one of the you have to really put yourself in the mindset of like the 1990s, and we thought of the world through the lens of DNA transcription in many ways, where you have promoter elements that would turn a gene on or off, and different sequences within DNA that are required to regulate mRNA expression. And so, when we started thinking about maternal mRNAs or mRNAs as regulatory nodes in gene expression, the gut instinct of the everyone was to look for sequences that are embedded within mRNAs that would then be repressive to the expression or stimulatory to the expression. And there was a lot of focus on the 3' untranslated region because the 3' untranslated region is relatively unconstrained by evolution because it doesn't interfere with the process of translation. So, there was so much work that went into identifying elements that would turn mRNAs on or off. And we found those. I mean, not me, but, the field identified these elements. But the thing that really was fascinating to me is that if you looked at, I really got interested in RNA stability very early on. And if you looked at RNA stability, the mRNAs degrade at all different rates. You got 25,000 mRNAs, they all degrade at a very different rate. It's not quantized, meaning you have mRNAs that are very stable and they always have the same half life. Every mRNA has that same half life, or intermediate, or somewhere down below. And rather, it's a continuum of half lives. And when we thought about that, it's like, well, how could individual RNA sequences give you such gradations in half-life if all these RNA sequences really do are bind, proteins, and now we know about microRNAs. If that's what they do, how are they actually giving these very discrete half-lives that we observe? And so we didn't really think that was the answer to why you would see these half-life differences between transcripts across eukaryotes. And that's not to say that these elements aren't incredibly important and don't contribute, but there have to be sort of general rules and mechanisms that are at play that really dictate overall half-life. And that's, what I pretty much started my postdoc and then eventually my lab on, was to try and figure out what those rules were.
Yeah, exactly. That's a great lead up to my next question about your 2015 work. Your lab found that the primary determinant of mRNA stability was actually the codon sequences, right, themselves. And that this was reading about it. It's very interesting. RNA stability is directly related to ribosomal speed of translation. Correct me if I'm anything wrong that I'm saying.
No,
Please
no,
feel
no.
free to interrupt me. But specifically, that the longer it takes a ribosome to translate mRNA, the proteins in the cell sort of recognize that stalling and then degrade the mRNA. And that is directly dependent on the concentration of
Hmm.
tRNAs for specific codons that are
Yeah.
present. And I think that's like a really interesting concept because I never thought about RNA stability as a function of translational speed. You know what I mean? Rather as this like inherent quality of RNA that all RNA degrades and it's just transient and the quicker the translation speed the better. I'm guessing that yeah, I'm again guessing that certain tRNAs are in higher concentrations than others. Since that finding, what has surprised you the most about the way that cells balance translational speed with RNA decay? Like, have you ever thought about the evolution of why this system
Yeah, I
emerged?
mean, it's fascinating because, I mean, let's take a step back. I
Mm-hmm.
mean, the first sort of clue that we had for this really does date back to work from the 90s, where we the field was trying to figure out how mRNA stability was regulated. And the hypothesis back then was that, well, there's going to be different pathways for mRNA decay. And so an individual mRNA will be a differential substrate for different paths. And a lot of work done in the 90s really showed that that, in fact, wasn't really true. There was really only one major pathway for mRNA decay, and at least in like model systems like Saccharomyces. And the so then it left, what is the explanation for why there are discrete half-lives? And so when I started my lab in 2005, I really wanted to figure out this fundamental observation that was known from the 1970s, where that it was known in the 70s that an mRNA that translates really well is very stable, and an mRNA that translates poorly is unstable. And we had to do a lot to sort of dissect out that mechanistically. And there were a lot of reasons why that was a difficult model to actually prove, because the world was thinking very differently in 2005, that mRNAs went to these discrete areas within the cell to be destroyed. These things are called P bodies, and they are areas where mRNA translation doesn't occur. And so one of the first contributions that my lab made to the field was that, in fact, we showed that mRNAs degrade while they're on ribosomes, that the process of degradation is co-translational, and actually gave a mechanistic understanding for why the process of decay really works the way it does. And just to back up, decay starts by removing the poly A tail, and then you remove the cap, and then you digest mRNA 5' to 3'. And that always was sort of a weird, why do you do that? Why does the cell go from one end all the way to the
It's
other
like,
and then back? And when you put it
yeah.
in the context of a translating ribosome, you can see that the decay process evolved around the process of mRNA translation, because the deadenylation is now limited. Tails are required for appropriate translation of mRNA. So the first step is to remove the
Mm.
tail, and you start to slow translation. Then you cut the cap off, you can't translate anymore. And then you degrade the mRNA 5' to 3' because you're following that last ribosome as it's moving through the body of the transcript. And that prevents the decay process from making truncated polypeptides. And so the whole thing has evolved, co-evolved with the process of translation in order to coordinate these two events. And so that was really kind of the first clue that something about translational elongation is very important. And just to go back.
Sorry, just
Yeah.
to, yeah, I'm actually really interested in that point. What kind of clued you in to the fact that we think mRNA degradation happens at the ribosome, like while translation is happening, as opposed to the
Well,
previously
it was really kind of it
speculated
was a
side?
very unexpected experiment. I was doing work, I still did work in the lab back then in the early 2000s, and we were doing a lot of purification of ribosomes because I had worked on this protein which I thought was very involved in controlling translation, and mediated a transition between translation and decay. And that's a protein called DHH1 or DDX6 in humans. And we were doing sucrose density gradients to purify ribosomes. And what we had this model in our head that the whole world had, which is that mRNAs degrade after translation occurs in a translationally repressed state. And when we were doing experiments with ribosomes, we always saw that there were decay fragments of mRNA associated with the
Mm.
ribosomes, and we couldn't get rid of them. And we thought that these were like artifact or purification, and that
Yeah.
we're just messing up. We don't know what we're doing. But we've
Oh,
been
that's
working
so
on RNA
cool.
for 15 years by that point, so we kind of knew what we were doing, at least in terms of manipulation. And and one day we just sort of had an epiphany where it's like, well, maybe the whole model is wrong. The way the field is thinking is wrong. That it's not occurring through a translationally repressed state; it's occurring on ribosomes. And we did a couple very simple experiments to prove that, and they worked. And it was the data were very convincing, and so that really opened up the floodgate to the eventual discoveries in 2015. And the really interesting thing about that discovery in 2015 is, at the time, before then, it was very dogmatic that the process of translational elongation is not regulated. it's always assumed that if there's any regulation in translation of an mRNA, it's always at the step of initiation because it's assumed that initiation is the rate limiting step. And that really comes from data primarily in vitro. And the idea that elongation could impact post transcriptional events was really quite heretical. But there were clues of this throughout the literature. And we had seen, the field had seen back like in the 90s when we were working on translational control in early development, that some of these RNAs, you could look at their patterning and see that they made no protein, they were translationally repressed. But then when you looked at them in more detail, they were on ribosomes. And so it made no sense. Like, how could they be loaded on ribosomes but not making protein? And there were sort of these little clues, several different observations from several different labs that saw the exact same thing. And it was always sort of ignored that maybe this is just, again, an artifact. But then in 2015, when we really showed that the mechanism was this idea of codon optimality, meaning ribosome speed is what dictates half-life, it really started to floodgate that elongation of translation. Ribosomes themselves are really the arbiter of gene expression events in the cytoplasm, that they, in fact, control most of the metabolism of mRNAs. And really, from an evolutionary standpoint, it's sort of fascinating. I'm getting back, long-winded answer to your question, but I'm getting back to your question about evolution and what I think about with this. Because if you look at all of the processes of mRNA degradation that we know about, so codon optimality is like the normal basal mechanism of decay. This is how most mRNAs respond to stability, how their stability is regulated. But there are other events. So, for example, if an RNA gets damaged by like oxidation, that can cause the mRNA to be the translation to stop, outright stop. And that now goes through a unique quality control mechanism called no-go decay. You have, what if the mRNA splices inappropriately? If it splices inappropriately, you might get a premature termination codon. Then it goes through non-cis-mediated mRNA decay. You might get another processing event where you lack a termination codon. So now the ribosome goes all the way to the poly A tail. That's called non-stop decay. And so all three of those mechanisms absolutely involve the ribosome sensing the quality of the mRNA. Okay. And now you have this normal pathway we talk about, which is sensing the quality, if you will, of an mRNA, how fast that ribosome can get through that mRNA. And so from an evolution standpoint, what you see is that the ribosome almost has, I don't want to say this would be anthropomorphic. It's like the ribosome's protecting itself, meaning that there's a lot of, I sort of think about it, that there's a lot of energy that goes into making a ribosome. That's over 300 different genes. It involves massive amounts of, ATP and nucleotide energy. It has to coordinate all three RNA polymerases. So there's a lot that goes into it. It's the biggest macromolecular machine in the cell and requires the most amount of energy. And so the biggest threat to a ribosome is, of course, the substrate it's bound to, the mRNA. Because if it gets stuck on that mRNA, well, what do you do? Okay, so there are all these mechanisms that have evolved essentially to get rid of that substrate, free up that ribosome. And normal decay is the exact same thing, that the ribosome is trying to move along. And if the tRNAs are just not in plentiful supply, the ribosome is moving too slow, and now it's being sensed by other machinery that then says, okay, it's time to degrade this RNA. It's outlived its purpose in many ways. So I really think it's the RNA stability and the different pathways is this elegant evolutionary dance between protein synthesis, ribosomes, the most ancient machine on Earth, and their utility to translate mRNA.
Yeah, that is so cool. Actually, I never thought about ribosomes through that evolutionary perspective. And honestly, just hearing about all of the experiments that your lab did to get to these conclusions, I mean, it's the most beautiful type of discovery where you're not necessarily looking for it and it kind of just happens and you
Very
realize,
serendipitous.
I've been doing this. Yeah, exactly. I mean, it's the honestly, in my opinion, it's the most beautiful type of discovery when you're not really expecting it. That is, yeah, it's really interesting. So, I mean, have you ever thought about how to? I'm sure you've thought about, what other practical knobs other than codon choice is now on the table to sort of tune stability. Like, do you
Oh yeah.
think that? Yeah,
Well,
what
I
are
mean,
your thoughts on that? I'm sure
I
this
think
is.
it's complicated because I really don't think about it necessarily as just being tRNAs. It's about ribosome speed, and there's lots of things that can influence
Yeah.
ribosome movement. So in higher eukaryotes, you have modifications of mRNAs, the presence of unique nucleotides that could dictate the ability of tRNAs to bind, for example, or for
Mhm.
proteins to bind that could then slow down the ribosome or interfere with its progress. Um, and so my gut feeling about this is that the machinery that is involved in monitoring codon optimality is used for lots of things whenever the ribosome slows down.
Mm.
And in fact, we're working on a story that suggests that even the rate of protein synthesis, meaning the rate of polypeptide synthesis, can impact that movement and feedback into decay. And so anything that you have to really think about it. There's different uh, when we're not talking about the ribosome stopping. When the ribosome stops, that's a bad thing, and that's called no-go decay. That you chop the mRNA, you cut it in half. We're just talking about subtle slowing of the ribosome, and that gives enough time for the decapping machinery to kind of sense the ribosome and say it's time to remove this mRNA. And there's lots of ways a ribosome could just slow down outside of just tRNA concentration. And I think that's where the field's sort of moving and seeing that there are other contributions to speed.
Yeah, absolutely. When people talk about RNA stability, it's interesting. You seem to have the perspective that the ribosome is kind of the main
Oh yeah.
character of this whole narrative, and that we should be looking at it through the perspective of what speeds or slows down translation from the ribosome's point of view. Again, being very anthropomorphic about this ribosome, but that seems to me,
I really think you have to think about ribosomes in that way. I mean, they are the most ancient
yeah,
machine, right, that we have in our cell. And
yeah,
the cell evolved around it in many ways, right? And I actually teach this sometimes to students, grad students, and they say, and I do this very tongue in cheek, but it's like the entire cell is there to do nothing but support ribosomes, right? Ribosomes are this selfish entity
hmm
that is supported by the cell, and the DNA and the nucleus and everything is there just to keep ribosomes making themselves. And in many ways, it's kind of true, right? It's like you have most of the genes are, well, not most, but a large component of the gene uh, in your body, genetic material in your body, is being used to support the production of ribosomes. And there are these, old
it's
observations.
true. It's
There's
true.
these old observations from like 20 years ago that you cannot separate cell growth and ribosome synthesis from each other. If cells grow, you make more ribosomes. If you limit ribosome synthesis, you stop growing. And so they're intimately connected to each other. The rate of cell growth and the rate of ribosome synthesis cannot be separated. And it really, it's kind of fascinating to think about that. Is that we're just these vessels for our ribosomes.
Yeah, I think a lot of people don't realize like bacteria have ribosomes. It's one of the few things that eukaryotic and prokaryotic cells
Yeah.
share. I think something like 80% of all of our RNA is our
Yeah,
RNA,
it's
right?
most of
Or
it is that.
even maybe even
Hmm.
more. Yeah, people don't realize that very small fraction of all of the RNA being produced in our body is actually is mRNA
Sure.
that's being used to create proteins. I mean, if you RNA is obviously used for the ribosome, you can maybe consider that a protein. I don't know, but functional proteins, I
Yeah.
guess. Yeah, it's an interesting thought that the cell was built to support a
Yeah, it's just a
ribosome.
it's just a fun thing to
Uh,
think whether it's actually true or not.
yeah, no, it's interesting. Yeah, I wanted to sort of transition into some of your more
Sure.
recent work. Your lab this year found that introducing an artificial poly A tail adapter
Mhm.
allows it to clip on to specific
Mhm.
transcripts, and that nearly doubles the protein
Yep.
yield in haploinsufficiency models. Can you kind of walk us through why you decided to append a synthetic tail rather than like inserting stabilizing elements in the coding region itself? Like, where did this kind of thought process come from? Maybe walk us through the rationale. Mature RNA naturally have poly A tails,
Yeah.
right? This is the post-transcriptional modification. And so is adding an additional tail supposed to be even more stabilizing? Maybe just walk me through your thoughts there.
Oh,
Would
yeah,
be
sure. I
awesome.
mean, it would be a long story, but I'll tell you. So in 2015, when we discovered codons can influence stability, this really gave a unique way to think about controlling mRNA stability. And there was this emerging technology that people were starting to talk about, mRNA-based medicines. Um, and the first author of that paper from 2015 went on to work for Moderna. He was one of the early employees of Moderna.
Hmm.
And we were thinking even at that time that if you are going to be making mRNA medicines, you really have to take into account codon optimality when making them, because knowing those rules
Yeah.
is going to impact dramatically how well the mRNA will be expressed. And so that author actually went on to design Spikevax. He was one of the lead designers in making Spikevax and is on the original patents for the COVID-19 vaccines. And so it was really kind of one of the first sort of entries that we had into the biotech space. And at the time, I was approached by a set of dads who were interested in technologies around haploinsufficiencies. Their daughter has a disease called Dravet syndrome, and Dravet syndrome is a haploinsufficiency where you have one of two copies of the SCN1A gene that's poorly expressed, and that leads to very debilitating epilepsy and a high chance of mortality by the age of 20. And so haploinsufficiencies are really tricky to think about and how to treat from a gene therapy standpoint, because you only need to increase the amount of protein by 2, right? You just have to just double it and go from 1x to 2x. And that seems like that should be relatively easy, but in practice, it's really not. I mean, first of all, most genes, human genes, are way bigger than the what we can deliver by an AAV vector, 4.7 kb. And then we have the added challenge that with haploinsufficiencies, and we don't know this for all of them, but one of the first that was described genetically was Rett syndrome. And Rett syndrome is a mutation in MECP2, and it's typically found in girls because the MECP2 gene is on the X chromosome. And so girls with Rett syndrome are mosaic. And so half their cells are expressing wild type MECP2, half are not expressing MECP2. But Huda had shown that she was the person who identified MECP2, and she had shown that some boys were coming into the clinic that manifest with similar phenotypes as Rett syndrome, and which was sort of shocking because, again, this is on the X chromosome. And when she looked at these boys more carefully, in fact, they had trisomy, so they were overexpressing MECP2. And so the kind of learning lesson from that was that underexpression of MECP2 is bad and it causes autism. Overexpression of MECP2 is bad and causes similar phenotypes. And so we now classify these as Goldilocks genes, where it's got to be just right. And so in terms of making gene therapy, well, this is the challenge that we have with haploinsufficiencies. Not all haploinsufficiencies are Goldilocks, but we don't know for a lot of them. There's about 300 that are classified in the human population, and most of them are ultra rare. And so the research into them is very limited. And with Dravet syndrome, we first went after this by using tRNAs. And we, in fact, were engineering tRNAs to read through stop codons, because about 20% of Dravet patients have stop codon mutations. And so the idea was to make suppressor tRNAs, these are called suppressor tRNAs that read through stop codons, put them into AAV vectors that would be a one and done treatment for these children. And then over time, so this was about 2018 when we started that work, over time, it became important to think about the impacts of AAV itself on potential treatments. There are lots of issues around AAV. There's manufacturing issues, quality control issues, and dosage issues. And then always the question, can we redose at some point if need be? And so I really wanted to think about, and I had talked to a lot of patient advocacy groups and such, start thinking about technologies that were non-viral that also could increase the expression of an mRNA. And importantly, not only so the early work we were doing was increasing the expression of mRNAs associated with nonsense mutations, but that's only about 20% of these patients. We wanted to make non-viral therapies that were also mutation agnostic, where it doesn't matter what the mutation the patient has. If we could raise the expression of the wild type mRNA, could we then overcome the haploinsufficiency? And with all that we know about mRNA stability and translation, it's really the poly A tail that is the key feature that dictates half-life of an mRNA and the overall expression of an mRNA. Okay, so now you got to put a little pin in that. All right, so now what does a poly A tail do? Well, poly A tail only does one thing. It recruits a protein called poly A binding protein, and has a very, non-imaginative name, but it's a very important protein, and it's critical for the translation of mRNAs. And but I had shown back in the 90s as a graduate student, I had shown that you could separate out the activity of the poly A tail from that protein. And I showed that you could basically put that protein anywhere in the 3' end of an mRNA, and it would act like a poly A tail. It would stimulate the translation. So now fast forward 30 years, and we're like, well, could we develop therapies with that idea that if we simply tethered poly binding protein to an mRNA, could you enhance its translation? And so we did this using these things we call poly A tail mimetics, where we simply have an antisense guide sequence, antisense to an mRNA of choosing, and then we've engineered onto that a poly A tail that can't be cut by the deadenylase. And so that can recruit poly A binding protein, keep it on the mRNA longer than naturally possible because of deadenylation that's normally occurring. And it just gives a little bit more expression of the mRNA. And that's all you really need with these haploinsufficiencies. So it's what I like about it is also it's non-viral. We would deliver these by by injection, direct injection, and then it could be very transient in helping deal with the haploinsufficiency, allowing time for new technologies to come online that could potentially be more one and done if that becomes possible and safer. So that was really kind of the story behind why we did it. And we're still in the process. Of course, it's very early days with that technology. We've basically shown it works in vitro and in mice, but we actually have to go in and do an animal model and cure an animal model of a haploinsufficiency, and then show so we can really get this to work in a safe and efficacious fashion.
Yeah, I mean, it's again very interesting that your graduate work is now coming to play later on in your career. My current thesis, my undergrad thesis, is heavily inspired by my PI's like postdoc work, and so he's been telling me that you're bringing up old memories, like all the old frustrations that came with this. You are now trying to tackle more, and so I can understand from like the student perspective of someone who's trying to reimagine their PI's
Science is
old research work.
science is a slow process, and I'll never
Absolutely.
forget. Back in the 90s when I was a grad student, I was thinking about doing a postdoc. And again, as an undergrad, I worked on RNA. So I was like, maybe I should do something different, right? So
Mm.
from 21, I was 21 when I first started working on RNA. And at the time, of course, everybody said you got to either switch model organisms or stay in the same model organism, but switch projects, right? When you do a postdoc. And I remember I had gone to a conference and I met this guy, and he's a professor, an older professor, and he said, I think that's a bunch of baloney, right? Because if you stay doing the exact same thing, or at least in the same area all your life, imagine what it'll be like in 20, 30 years, where you now have this huge knowledge base of work in that field that others won't have. And you can rely upon old observations that you have and old ideas and everything else. And it's actually true. In many ways, we are doing that, and we still continue to do that to this day. And it's very beneficial and it's nice because then you spend your entire life working on a set of problems, and as long as it keeps you interesting, it's like slowly over time, you learn new and fascinating things about that problem. So, to me, I never find this job boring at all.
I 100% agree. I mean, especially if the problem is one that you're innately very passionate about and curious about, and if it drives you, then, what is the need to do something different just for the sake of doing something different?
That's right.
Yeah, I mean, you kind of touched upon some of the more entrepreneurial side of RNA biology and RNA research. It's hugely embedded with
Yeah.
biotechnology companies now, right? You co-founded biotech startups like Tavard Biosciences. Is
Mhm.
that how you pronounce
Yep.
it? That's pursuing tRNA-based treatments for genetic diseases. And then I'm not sure if I'm going to pronounce this right. Wive RNA.
Yeah, that company has no longer exists, so we don't have to talk about that one. But Tevard's still,
Sure
yeah,
thing.
Tevard is still going strong, and I can go in there tomorrow, and they're working predominantly on muscular dystrophy, Duchenne's muscular dystrophy, and cardiomyopathy, and this is a company that was started with those two dads and Harvey Lodish from MIT, and they still want to pursue treatments for haploinsufficiencies in the brain. So it's been an interesting process because we went from that company being my lab, one person in my lab, to now, it's now in Boston in the Eli Lilly's new building. And, we're close to going to the clinic. And that's really fascinating to see as a basic
Yeah.
scientist that, these ideas that you had very early on and, you never, we don't think about it like that. They make for interesting puzzles and might make, cool publications, but actually they could make a difference in people's lives and really, be important treatments and potential cures for patients. And that's very humbling in many ways because it brings back, kind of what I consider the social contract of scientists such as ourselves that are really funded through the American taxpayer that, we do that because it's a promise. The American taxpayers are wanting us to return upon their investment. And we tend to get lost in our thoughts about this, how we have to go from the next Cell, Science, or Nature paper and get our grants and tenure and all this stuff. But really, we're not here for all that. We're here to bring these discoveries to the American people and to make their lives better. And that's not just lip service. It's really important for scientists to think about because if we, another thing that I do a lot of more recently is advocacy and government advocacy. And I think a lot of scientists, we've lost that ability to communicate what it is that we do to the public. And it's one of the reasons why I think there is so much distrust of science and scientists in the United States right now. And why, some of NIH and the mRNA medicines community are really under systematic attack because we've forgotten that mission of, that we really do work for the American people and we owe it to them to explain better what we do and how what we do improves their lives.
Beautifully said. The academia system is, I mean, the goal of basic science research is to set the stepping stones for these therapeutics, diagnostics, whatever, to eventually make its way to market and reach patients. But the way the system is set up doesn't necessarily incentivize for that, right? It incentivizes for, like you mentioned, publications, tenure, kind of rising up the ladder within that bubble. And I think that's why there's so much IP just like sitting around in universities totally untouched. I think a lot of people in the science community don't value, and correct me if I'm wrong, but my impression is that they don't necessarily value that aspect of it, of commercialization, of taking it to market. And I think you have had very unique experiences where you've spent a lot of your life in academia, right? But you're also very deeply integrated into, these products and actually bringing them to patients. And maybe that's because of the nature of this field. Maybe it's because, like I mentioned from the very beginning, that RNA biology, RNA therapeutics are very hot right now, but nevertheless, it's really seeing a great impact in patients. Maybe you can speak a little bit to that and maybe your experiences. I mean, I'm curious because, it's not easy to start a biotech company,
Yeah.
right? And a lot of scientists are not necessarily built for that. There's so many different aspects of it. You really, my impression of it is that it's less of a, I would say less of like you go away from the science a
Yeah.
little bit, and it becomes more about how do you scale this? How do you, how, when you're capital strained, how do
Yeah.
you create enough runway to scale the technologies, to test it in patients, to then get more money? And a lot of it comes with those sort of technical barriers and a lot of the financials as well. So I'm curious about maybe some of those challenges that you've faced and how you've gone about addressing them, whether you've gotten help from others, whether you have found that you've had to learn a lot of this yourself, and maybe just some of the
Yeah.
challenges, but also some of the rewards.
Well, you
Yeah.
learn a lot of it on your feet. And I mean, the truth
Mhm.
is, I think most scientists don't get into biotech development because they simply don't know what to do or how to do it. And you do need a strong
Mm.
support network to do this. And you can't ignore your primary job, which is your lab. And you have to balance those two things. And in my case, I've had, great mentors, Harvey Lodish, who, we started Tevard with as, one of the big biotech leaders in the world, in Boston in particular. And he's been a great mentor to watch how to navigate a lot of these complexities. And the other side of it is that, you as a scientist, even when you have discoveries that come out of your lab and you want to spin them into tech, you don't necessarily have to be the one who leads that company, right? And I don't do that. I
Yeah.
mean, the CEO and CSOs of Tevard are not me. I'm just a strong voice there, and that also has consequences to it. I can talk about in a minute, but you
Mm.
really have to find what it is that you want to do, how you want to be involved. And in my case, and in the very early days with Tevard, I was flying to Boston once a week, and I would get up at 4 o'clock in the morning and be
Oh.
there and work all day and then fly back and be in my bed at midnight. And it was exhausting, but we did that for a couple of years, and then the company grows and it starts to take on a life of its own. And the one of the hardest things as a scientist that I found was the missions between the two, or your lab and a biotech spin out, are very different. And there's a lot of times, we were doing work at Tevard that'd be like, hey, that's cool. That's a cool observation. I'd love for us to follow that up. But you you have to listen to that inner voice that says, no, we don't do that. We just keep moving forward to go toward the mission, which is, to develop a therapeutic for a particular disorder. And that also has been beneficial to my lab, though, because I've some of that, as we've learned things that have cropped out of peculiar observations from experiments that have been done at Tevard. Some of those have turned into projects in my lab, my research lab at Hopkins. And so it's like you
Oh.
can use both to kind of learn from each other,
Yeah.
but you have to balance that. One, the academic lab can sort of play all over the place and do things, where the biotech lab has to be laser focused on getting a product to the clinic to get it into patients' lives, to improve patients' lives. Um, and so those are some challenges, and of course there are other challenges, like raising venture capital. That was a very difficult thing, and I probably had well over 50, 60 different calls, explaining the science to people and getting investors interested in the technology. And it's a lot of work. It's a lot of work in addition to what you normally do in a given day. But then the most rewarding aspect of it, though, is when you go and you get invited to these small patient advocacy group meetings, and then you really
Mm.
meet the people that you're trying to help. And I'll never forget it. I went to one of these meetings in Nashville, and they had a little mixer after the talks and stuff, and I'd given a talk, and I talked a little bit about what Tevard does. And this woman came up to me, and she was just a normal everyday person, and she rolled up her sleeve and she had on her forehead a tattoo. And the tattoo was the genetic diagnosis of her child, which is basically showed what the mutation was: amino acid XXX
Mm.
going to a stop codon. And she's like, could your therapy help with this mutation? And I was like, you have that tattooed? And she's like, well, I don't understand it, but everybody I see that, like you, I show it to, and they know what it means. And I never want to be able to forget it, and I want to say it accurately every time. So she had it tattooed. And it's like, wow, the dedication of a mother to have her child's diagnosis on her arm so that if she runs into a random scientist at one of these meetings, she can ask, could your therapy help my kid? And I thought that was just like mind blowing because it's like, well, this is why we do this. We do this because there are, families out there that are really struggling, and they don't care about all of our tenure and our grants and our, science nature papers. They want these things for their kids. And that really changes the way you think about the scientific process. That, the most rewarding thing for me will not be a legacy of, you know, papers on my CV, is if I can get something into a patient population that does help people. I mean, that well, I can retire at that point because I don't need to do anything more. I'll be happy. So,
Oh, that's beautiful. Yeah. I think that, the beauty of academic research is what you just mentioned, that you have the freedom and the curiosity to explore different areas that interest you. You find a question here, you can explore that. But I mean, there's also the beauty to the whole biotechnology industry, right? And what it can do for patients. And that story you just mentioned, like that, you know, that has the power to actually affect millions, if not billions of people, depending on what you're going after. And yeah, that's absolutely beautiful. Yeah, I think I'll maybe end this conversation with the fact that there's so much excitement in this field and how it can affect patients, right? I mean, RNA therapeutics is exploding in the possibilities, and you have been at the cusp of that. Is there an RNA therapeutic or an application of RNA that you personally find most
oh
exciting right
yeah,
now?
absolutely. And I so it's ironic. So because I wrote an editorial that'll come out today, in fact, in the Wall Street Journal, about the future of
Oh,
mRNA
wonderful.
medicines. And the one of the most exciting things that I've seen was a paper that came in 2022 from a group at Sloan Kettering where they had used an mRNA. So they had gone into a cancer, pancreatic cancer patient, PDAC, and they sequenced the tumors and then discovered the neoantigens. And then they stitched those neoantigens together as an mRNA and then inject that mRNA back into the patient. And when they do that, so first of all, pancreatic cancer is like a 99% mortality rate within 6 months to a year. One of the most deadly cancers that we still haven't really made any advances in decades of work. And when they did this neoantigen approach, 50% of the patients responded and their tumors regressed. And they had a follow-up study a few years later. Those patients are still around. So here we have a technology, 50%. That's not great, but it's better than what it was. And it really, you have to take a step back. I'm a little older than you. And when I was young, people were terrified of childhood leukemia. And childhood leukemia had about a 95% death rate in the 1940s. I'm not that old, but in the 1940s, it was almost like pancreatic cancer. If your child was diagnosed with
Hmm.
leukemia, palliative care was the best you could do. Every decade, we got a little bit better with those numbers. Every decade, a little bit better through hard slog work in research and development, to the point where by 2025, children don't die of leukemia anymore. It's like 95% survival rate. So, 90% death rate to 95% survival rate, complete 180-degree reversal in that. And wow, can you imagine? Might be where we are with pancreatic cancer or some of these other, incredibly scary cancers like glioblastoma or triple negative breast cancer, where the mRNA based platform would be explored and amplified in a research fashion to really get us from, okay, we have 50%. Could we get to 60? Can we get to 70, 80, 90? Could we have cures? So that by the time our grandchildren do a podcast like this, they'll be thinking, well, remember back when pancreatic cancer was scary? Now it's just nothing. That would be
Yeah.
fantastic. So to me, that's the most exciting thing that I've seen in science in a long time, where we're really on the cusp of these incredible biomedical, miracles. And we just need to continue the investment into this space and talk to our leaders in Washington and our friends and neighbors and really explain in clear, simple words why it is we do what we do. And that analogy of childhood leukemia is one that people should remember and share, because it's a true American success story of innovation.
Absolutely. You have a beautiful way of explaining these concepts in a very clear, understandable way, but also in a way where the person listening may not necessarily know anything about pancreatic cancer, may not know anything about leukemia, but they're listening and they're realizing, wow, this is the impact of science. This is what science can do for patients. And I'm totally with you in that if a legislator were to hear that message, no one would be questioning the value of the money that goes into
Well,
science and
feel
research.
free to use it and call your representatives tomorrow.